So we're going to introduce the topic of decision making. Well, critical to making decision is being able to not only ascertain what options there are, but also to give value to any decisions that are made. So for example, if we take our mouse, every moment of the day it faces different options, different choices. There are two key questions then for this mouse. What are the values of the available choices? So for example, it might face the choice of, do I go forage for food or do I stay safe at home? Or while foraging, it might face a choice, do I go to the place where I found food yesterday or do I try a new place? The second type of question is the flip side of that. In terms of the valuation, what are the costs to each choice? So if I stay home, if the mouse stays home, then the cost is it doesn't get any food. If it ventures out to find food, a potential cost is being eaten. So every moment of the day, animals are faced with life and death decisions. So from an evolutionary point of view, natural selection would favor brains that are making decisions in a wise way. Today we're going to introduce you to some of the research presented in a book by Reed Montague, and that's his name there, Reed Montague. And the book originally was titled Why Choose This Book? But I believe the publishers, or he with the publishers, have changed it. I'm not sure what the new title is, but it was formerly known as Why Choose This Book? Now, Professor Montague, in the beginning of the book, discusses the evolutionary context of decision making. And here's a quote from his book. So listen carefully. He says, "In order to survive, mobile creatures must be able to value the resources available and the choices they can make to acquire those resources. An animal that moves in one direction forgoes resources it might have acquired going in another direction. For the same reason, even eye movements represent important choices. Seemingly small choices like eye movements or where to step next may seem irrelevant, but they add up vitally over time, for the good or for the bad. Nature's economic realities have selected for creatures that can value their intended actions quickly and accurately." So, as he goes through the book, he defines a couple of terms that will help us sort of connect up the brain research with our everyday sort of psychology concepts. He defines a goal as being a desired state, a future state whose representation can be used to guide performance to reach that state. All right. Now, as the mouse, for example, it might have the goal of filling its belly. You see, it goes out into the world, and it's going to be foraging and nibbling on things, trying things, and so on. Well, if it tries a little bit of that stuff and it doesn't taste good or whatnot, it won't continue eating it. Right, so there are error-correcting signals, and we'll call these guidance signals. So guidance signals help the decision systems adjust from deviations from the goal. And Professor Montague wants to call these sort of a model for the concept of desiring something. So we have a goal, and we're out there trying to satisfy the goal. Since we have the goal in mind, we can compare our current state to the goal state, and any deviations from the goal state then serve as an error correction signal. All right. Okay. He also defines wants as stored changes in the value of an ongoing state. When the change is positive, so some kind of change is happening in the brain that has a positive value, like the effects of cocaine, for example, the stored state becomes a goal state that is wanted. Right? So the mouse, if it were a cocaine user, would want that high that it got from using cocaine. So we have the concept of a goal, concept of error signals or guidance signals that help us reach the goal so we can sort of correct our performance to better match the goal. And we can think of that as sort of desire, right? And then the wants are those things that are stored in the brain with high positive value, and these are things that the goal states are intended to achieve. So, in order to figure out how the brain is assigning values to things, researchers started to look more closely at the dopamine system. It was long known that the dopamine system seems involved in pleasure, for example. However, some of the old ideas about dopamine are being overturned with new research. The dopamine system is affected by drugs like cocaine and amphetamine. These drugs increase the levels of dopamine at synapses, and then that is correlated with a euphoric state. So again, there is a pretty good correlation. Chemical compounds that increase the amount of dopamine in synapses, for example, will tend to produce pleasurable states. Rats will work very hard to give themselves injections of dopamine into the brain. Of course, they don't themselves do the injection, but if they had micropipettes inserted into the brain where the dopamine neurons are, and I'll show you that in a moment, and there is some setup in their cage where if they push a bar, they push something, there's a little squirt of dopamine, they will work hard to learn how to get the syringe to squirt dopamine into their brain. Another way of doing that is to put electrodes into the dopamine neuron systems and to just stimulate the dopamine neurons. So they release dopamine as a neurotransmitter. Again, rats will work very hard to learn how to do that. It is pleasurable. So here is the dopamine system. There are two main areas of dopamine-containing neurons. One is called the substantia nigra, the orange ones, and then the ventral tegmental area, the green location. Both of these locations have dopamine-containing neurons that send axons through this pathway called the medial forebrain bundle up into frontal cortex. And this is true of humans as well. This is a cross-section of a rat brain. Now, the substantia nigra neurons are the ones that are implicated in Parkinson's disease. So Michael J. Fox and others have a deterioration of dopamine cells in substantia nigra. So the dopamine, that pathway is, destruction of that pathway is associated with some movement disorders. But we're going to be focusing on another dopamine pathway, the ventral tegmental area, which runs up through another region of the brain called the nucleus accumbens. I'll show you that in a bit. And then the pathway continuing on to the frontal lobe. It's this pathway, the green one here, that is thought to be a valuation system. So the dopamine system is going to be sort of keeping track of the positive or negative values associated with objects and actions and goals. So because of that, it's a critical component of our decision-making machinery. And it would be classified in the emotional category rather than the deliberative reasoning category, which makes use of frontal cortical areas. So this is an emotional system that is giving value to the things that we do and the things that we interact with, so that we can make better decisions in the future. Now here's an example of a rat working hard to deliver either cocaine or amphetamine, for example, into its brain. If it pushes the lever, the syringe squirts a little into its brain. It doesn't have to be, it could be systemically, but it'll affect the brain. Here's a graph showing what happens when you ingest cocaine. The blue one here would be baseline dopamine levels. And with the introduction of cocaine, you'll see dopamine levels dramatically increase. So cocaine is influencing dopamine concentrations at synapses. Now here's a human brain, again showing some of these pathways. The VTA, ventral tegmental area, is the origin of these dopamine cells. And here is this pathway, the medial forebrain bundle, this pathway going up to this red splotch here, the nucleus accumbens. And again, if you stick an electrode in here and stimulate it in humans or rats or whatnot, it is a pleasurable experience. Of course, the neurons continue to send axons up into prefrontal cortex as well. And so it's this interaction between this emotional dopamine system and the frontal cortex that situates the dopamine system so that it can influence decision-making that is sort of ongoing in the frontal area of the brain. Okay, so I mentioned before that new research by Montague and others is revising some of the old view. It's no longer believed that dopamine is just representing the amount of pleasure that something gives. It's more subtle and interesting than that. Okay, so what is the nature of this new research? First of all, then, we have to describe the experimental setup. So you've got the rat, you've got electrodes inserted into its brain. Just a moment. Electrodes inserted into its brain in the dopamine containing areas. All right. So the medial forebrain bundle has the fibers, the axons, and then the ventral tegmental area, or the nucleus accumbens, would have cell bodies. So we've got electrodes inserted into the brain. We can record and stimulate, and we can just monitor the dopamine neurons when different things happen to the rat. So for example, if the rat is just hanging out and a light goes off, so a light turns on, and we're measuring the dopamine firing rate, so the rate of action potentials, it's likely that you won't see a change in the firing rate of the dopamine cells. So if before the light went on they were firing at a certain frequency, when the light turns on, that frequency doesn't change much. However, if we come along and give a little squirt of juice into the rat, you'll get a spike in dopamine activity. So an increase in the frequency of action potentials. Now in the old days that would have been interpreted, aha, see, the rat is liking that, right? You squirt a little tasty juice, good stuff, feels good, pleasurable state. Dopamine cells are simply correlated to how pleasurable something is. But that's not quite the modern view. To understand the modern viewpoint, we're going to do a simple classical conditioning experiment with the rat. So what we're going to do here then is we're going to flash a light and then give it some juice. And we're going to pair those. Light, juice. Light, juice. So here, this is a classical conditioning paradigm where you repeatedly pair the conditioning stimulus and the unconditioned stimulus. In this case, the light was originally a neutral stimulus, and the juice was an unconditioned stimulus. That was originally something favorable, something good, something rewarding. Well, pretty soon, right, we know from classical conditioning research that pretty soon the rat will begin to anticipate, when it sees the light, it'll anticipate this reward of the juice. Right? Now in Pavlov's situation, the dog would start salivating, right? Just at the smell of the food, for example. And in the foot shock experiments that we talked about before, you pair a sound with the shock. Pretty soon the rat starts to become afraid of the sound alone because it predicts the shock. So in this case we've got light, juice. Now here is the interesting thing about the dopamine neurons. This is what happens to the firing rates of dopamine. So to see the setup so far in graphical terms, we can use this slide here. When the rat is just hanging out, minding its own business, and if we were to record from the dopamine cells, we'd find some baseline level of activity. Here the two little slashes means that there's some frequency of action potentials, and we're going to call that baseline. Now, as I just said, if a light comes on and the light has no significance, what researchers found is that you don't get very much of a change in dopamine activity. And psychologically, we could say that since the light has no significance, it's not particularly pleasant for the rat. But if we come along and squirt some juice, then you see an increase in dopamine activity, higher frequency action potentials going down that axon. And again, the old way of looking at that was, well, this is just correlated to the pleasantness of the current state. So because the juice is pleasant, the dopamine activity is the neural correlate of that. Now, the modern research that is going to change this view, change this interpretation, is as follows. What you do is you pair the light with the juice. So let me go back a second here. So we're going to just give the light, flash the light, and then a squirt of juice. Light, juice, light, juice. Now remember, originally you get dopamine activity when the rat is getting the juice. But now look at what happens when the rat is trained. When you have a classically conditioned rat, so that it begins to expect juice after it sees the light, then the dopamine activity is greatest when the light comes on. And it falls back to baseline when the rat gets the juice. Now the juice is still pleasant for the rat. So you see, this is how the old view of the significance of dopamine activity has been changed now. In the old view, the idea was, well, juice is the thing that's really pleasant, right? So we would have expected a burst of dopamine activity when the rat gets the juice. But it turns out when you classically condition the rat, now you get the burst of dopamine activity when the rat sees the light. Now here's the current view of what dopamine is doing here. The dopamine system doesn't seem to be tracking just the state of pleasantness, but rather it's tracking something more like the expectations for the animal. If something better than expected comes along, then you get a burst of dopamine activity. But if something comes along that was expected, the dopamine activity will not respond. And now we can use that interpretation to understand the original situation with the rat. See, if the rat is just hanging out and the light comes on, well, that's not better than expected. The light comes on, didn't have any significance. But if the rat is just hanging out and gets a squirt of juice, well, then that is better than expected, right? Because it was not expecting to get a squirt of juice, and now all of a sudden it's got juice in its mouth. That's good. Now look at what happens then when you train the rat. Light, juice, light, juice, light, juice. Now the light predicts the juice. So if the rat is just hanging out and the light comes on, that's better than expected because the light predicts the subsequent juice. Notice the juice is expected because of the previous training. So dopamine activity falls back to baseline. It's the light now that is the better than expected stimulus in the rat's environment. So dopamine activity is not tracking just pleasantness, but it's tracking a condition of the animal that's better than expected. Whatever stimulus is better than expected, you'll get a burst of dopamine recording that. And that signals the brain that, that this, whatever that is, that stimulus, is something that the brain should keep an eye out for. So dopamine activity is not correlated to just how pleasurable something is, because the juice is pleasurable here. But it seems to be tracking this idea of something better than expected. Something's got greater value than expected. Theoretically, if we, prior to the light going on, we had some other stimulus, a bell goes off, So if we train the rat bell, then the light, then the juice, what we would find is that dopamine activity after training would spike when the bell was heard and go back to baseline for the light and then the juice. So there's a transfer of valuation in this system. So stimuli that can predict later good things, they become highly valued by the dopamine system. Now Montague introduces a term here that's sort of useful, and he introduces the concept of a value proxy. So after pairing the light and juice, the light becomes a value proxy for future juice, and so can allow the brain to make better decisions about the future. The increased dopamine activity tells the rest of the brain to treat the light as though it carried the same intrinsic reward as the juice. Now the animal can be on the lookout for the light as it predicts the juice. And this chain can be carried further back. So as we said, if we now always have a sound before the light, before the juice, this value proxy can go down the line. The value can be transferred to the first stimulus in a sequence. And this is going to give an animal then more time to find the ultimate goal state, which would be the juice. If you can know how to get the juice 3 or 4 steps in advance, that is helpful in getting the juice.